All right, let's talk about some other organelles that use membranes. Endomembrane system. So endo means inside. So we're talking now about the membranes of the cell other than the plasma membrane. So the plasma membrane is on the outside, controlling what comes in and out of the cell, and the endomembranes are membranes that are inside the cell. So that endomembrane system consists of internal membranes and organelles in eukaryotic cells. They work together to modify, package, and transport lipids and proteins. Included in this system are the nuclear envelope, lysosomes, vesicles, endoplasmic reticulum, and the Golgi apparatus. Now they're including the plasma membrane, but we'll talk about that separately. All right, lysosomes are more or less a vesicle. These are in animal cells and they contain digestive enzymes. So enzymes are proteins that make a reaction happen, and these can break down large biomolecules and worn out organelles. Endoplasmic reticulum is made up of interconnected membranous sacs and tubules, and endoplasmic reticulum is connected, it's continuous with the nuclear envelope. It can produce proteins, and that's the rough ER is producing proteins or synthesizing, and then the smooth ER synthesizes lipids. The hollow portion of the ER is called the lumen or cisternal space. We're going to use this word lumen again throughout the semester, referring to the inside, the open inside, right? For example, the inside the tube of your digestive system, we call that the lumen, right? That where the food is, right? Would be the lumen. All right, and as we said, the membrane of the ER is continuous with the nuclear envelope. All right, rough ER. Ribosomes are attached to the rough ER. That's why it's rough, and you can see the little dots in an electron micrograph. So the ribosome is attached to the cytoplasmic surface of the endoplasmic reticulum, manufacture proteins. And these new proteins can be modified by folding them or adding some side chains in the lumen, the inside of the rough ER. Modified proteins are either incorporated into cellular membranes or secreted from the cell by way of vesicles. And these are going to be hormones and enzymes, adrenaline, things like that, things that are going to be excreted from the cell. All right, we've also got some phospholipids being produced. Phospholipids are modified proteins not destined to stay in the rough ER, reach their destinations by way of vesicles. So there you've got a little vesicle blebbing off. That is a transport vesicle carrying stuff to another site. In this case, it's going to the Golgi body. Smooth endoplasmic reticulum does not have ribosomes or has few. There it is, it's all smooth. And it synthesizes carbohydrates, lipids, and steroid hormones. There's also some detoxification of medicines and poisons and storage of calcium in the case of muscle cells. So the Golgi apparatus is sort of our pack and ship. And so these lipids and proteins that have been produced in the cell are going to need to be sorted, packaged properly. Some of them are going to be sent out of the cell by way of vesicles. The Golgi looks like layers of flattened sacs or membranous sacs. Here's a vesicle that is reaching the plasma membrane, and then the the outer part of that vesicle is membrane, and it's just going to bleb into the plasma membrane, and the contents of that vesicle will be released to the outside of the cell. The receiving side of the Golgi is called cis. The opposite side is the trans. So there's vesicles coming from the ER. They're landing on the cis side. The particles going out are the trans side. Transport vesicles from the ER fuse to the cis, and their contents empty into the lumen. When these vesicles land on another membrane, think of some detergent bubbles in the sink as they kind of bleb into one another, so that those molecules that are forming the membrane of the vesicle are just going to bleb into the membrane of the Golgi body or into the plasma membrane. Right? So the Golgi can modify and sort. It can add some chains of sugar molecules to the molecules that have arrived. Then we've got the cytoskeleton, which is a network of protein fibers. It maintains the shape of the cell. It may hold organelles in a particular place. It can allow movement of vesicles within the cell, and in some cases it's going to enable the cell to move. So that's the cytoskeleton. Is made up of 3 different kinds of filaments, and we'll get to that in the next mini lecture. So this is the last slide of the cell mini lecture C.